In Chapter 15, we're going to talk about how microorganisms cause disease. So these two definitions of pathogenicity and virulence are really important, so make sure you know these for the exam. Pathogenicity is the ability to cause disease. And as we've learned before, not all microorganisms are pathogens and cause disease. Virulence is talking about how severe the disease is. So for example, if we compare the flu virus with a cold virus, the flu virus is more virulent than the cold virus. And that's because you get sicker with the flu than you would with the cold. So in order to cause disease, pathogens have to enter the host. They have to adhere or stick to your tissues. And then a lot of times they will penetrate deeper into your body, and they also need to be able to evade your immune system. And then they will cause damage to your tissues, and that's what causes the disease. Virulence factors are just microbial structures and products that allow them to do those above four things in order to cause disease. So we have some different types of virulence factors. We'll look at adherence factors, which allow them to stick to our cells, invasion factors that allow them to spread through our body, and then we'll look at some factors they use to escape our immune system, and we'll look at several different types of toxins that can damage our cells and result in disease. This picture here is a good overview of what we're going to be talking about in this chapter. So we're going to start by talking about the portals of entry, so how the microorganisms get into our body, and then we'll look at how they adhere to our cells, and then next we'll look at how they can penetrate in deeper and invade our immune system, and then how they can damage our cells and cause disease. And then portals of exit is how they're going to get out of our bodies and then go and infect the next person. There are three different portals of entry for pathogens. So the first one is they could get in through the skin. Second, they could enter through mucous membranes. And the third route is they can enter through what's called the parenteral route. And that's spelled Parenteral. So parenteral just means that it's somehow been injected underneath the skin. So that could be a needle stick. It could be maybe a scalpel blade from a surgery that introduced bacteria underneath the skin. It could be a dog bite, insect bite. So anything that just injects it into your body and gets it underneath the skin, that would be the parenteral route. And pathogens are very specific in how they get into your body. So if they're in the wrong spot, they won't be able to get in and cause disease. So for example, the flu virus, if you get that on your hands and then you wash your hands, you won't get the flu. And that's because the flu virus can't enter through the skin. But if you didn't wash your hands and then rubbed your eyes or your nose, or maybe you ate something, then it could enter in through those mucous membranes. This is just a table just showing you some different portals of entry for different pathogens. So here's some mucous membranes, microorganisms that get in through the respiratory tract, GI tract, genitourinary tract, and then also has some that get in through your skin, or maybe they'll use a parenteral route where they're injected and end up below the skin. So the number of microorganisms actually is really important. So the more they get into your body, the better chance you're going to have of developing the disease. So for each microorganism, we have an infectious dose 50. So that's just the number of pathogens that will infect 50% of a sample population. So a good example of infectious dose 50 would be for E. coli. So for E. coli, it would take ingesting 1 million E. coli bacteria to make you sick and get diarrhea. But for another microorganism, for example, Cryptosporidium, which is a protozoan that also causes diarrhea, you only have to ingest 10 of those to get sick. The lethal dose 50 is just the dose that's going to kill 50% of a sample population. So this graph is just showing you the infectious dose 50 for two different strains of a microorganism. So you can see that strain A has an infectious dose 50 of 3,000 organisms. So that means half of the population, if they ingest 3,000 of those microorganisms, then they will get sick. Strain B has a higher infectious dose 50, and that's at 5,000. So strain A would be the worst one to be exposed to because it's going to take fewer of those microorganisms to make you sick. We also have some other virulence factors. These are called adherence factors. So these will allow the pathogens to adhere or stick to your cells. So we talk about adhesins that are on the pathogen that will bind to host cell receptors. So that could be part of the glycocalyx, which in bacteria, that would be their capsule. Also in bacteria, we have the fimbriae, which we'll look at on the next slide. And then remember that bacteria can also stick to things by forming those slimy biofilms. So here's a picture of some adherence factors. So up on the top, we've got the fimbriae. So remember, those are little short protein filaments that stick out, and they allow the bacteria to stick to our cells. We also have capsules in the middle picture. So the capsules are made out of polysaccharides, and they're sticky, and so they will allow the bacteria to stick to our cells too. And then the bottom picture is showing the viruses with their spike proteins, which will bind to receptors on our cells and then allow them to get inside. Okay, so there's a couple of different factors that will help bacteria invade a host, avoid the immune system, and then obviously increase their virulence and be able to cause more severe disease. So we'll take a look at capsules, some cell wall components, some enzymes that bacteria make, antigenic variation, and then how they can also rearrange the host cell cytoskeleton. Bacteria are able to resist being phagocytized by several structures that they have. So one would be capsules. So over here in the picture on the top right, there's a capsule there. Remember, those are very slippery and it's hard for the phagocytes to grab onto them. And then we also have in our Streptococcus bacteria, they do have a capsule, but then also coming out of the capsule, they have these M proteins. And that actually prevents the complement proteins from binding to the Streptococcus bacteria. And so then they're not opsonized and the phagocytes can't grab onto them. And then on the bottom left, we've got some orange staining tuberculosis bacteria that are actually living inside the macrophages. So the mycolic acid that's in their cell wall, remember, is pretty waxy, and that actually prevents the digestive enzymes from the lysosomes from digesting those bacteria, and so they're able to live inside the macrophages. Bacteria will produce several different enzymes that will also increase their virulence. So we'll look at a number of these on the next slide. And then I just wanted to point out on the bottom the IgA proteases. So those are enzymes produced by the bacteria to destroy the IgA antibodies. And remember, those antibodies are secreted across mucosal surfaces, and they will bind to the bacteria and prevent them from being able to bind to our cells. So they will destroy those, and then they can go ahead and stick to our cells. So here are some more enzymes that help the bacteria evade our immune system and allow them to spread to other parts in our bodies. So if you look on the bottom left picture, we've got some bacteria that secrete an enzyme called coagulase, and that actually causes a blood clot to form around the bacteria. And the purpose of that is the clot will hide the bacteria from our immune system. So the bacteria hide inside the clot, and then when the white blood cells go away, they secrete another enzyme called kinase that will then dissolve the clot, and then they can spread to other areas in the body. Streptococcus bacteria produce streptokinase, which was a drug that used to be used to treat blood clots. So if you had a heart attack or stroke, they would actually give you streptokinase that was made by the bacteria to break up the clot. And then the picture up on the top right, we have two other enzymes bacteria can produce. So one of them is called hyaluronidase. And hyaluronidase is an enzyme that will break apart the polysaccharides that hold these epithelial cells together, and that'll allow the bacteria to slip in between those epithelial cells. And then when they hit the basement membrane here, which is connective tissue made out of collagen, they make another enzyme called collagenase that will dissolve the collagen. And now the bacteria have broken through that epithelial barrier and they can go deeper into your body. Another way that microorganisms will avoid the immune system is some of them can do antigenic variation. So let's take a look at this on the next picture. So an example of antigenic variation is done by a protozoan. It's called Trypanosoma brucei. So this causes African sleeping sickness. You can actually see the trypanosoma over here in the top right, and it's going to be transmitted by the bite of a tsetse fly, and it'll get into your bloodstream and then make its way to your brain and then start damaging your brain, and it actually disturbs your sleep patterns. So that's why they call it African sleeping sickness. So what this protozoan does is that it keeps changing its antigenic coat. So you can see from this graph, the variation A of our trypanosome will increase in number, but then it starts to fall off as the immune system catches up and starts making antibody and killing off variation A. The trypanosoma seem to sense that, and then they change their antigen coat, and now they look different to the immune system, so those antibodies no longer will bind, and now the trypanosoma can take off again, increase in number, until the immune system produces new plasma cells that recognize this new variant, and then antibodies start being produced and numbers go down, but then they change their coat again, and so they always are one step ahead of the immune system. So the advantage of this is that the immune system will never get rid of this parasite because it keeps changing how it looks, it keeps changing its antigens, and so the immune system can never catch up and fully get rid of the parasite. Okay, another thing that some bacteria do is they will produce some proteins called invasins. And invasins are going to alter the microfilaments, which are made out of actin, so they're part of the cytoskeleton. So the picture on the bottom left shows some salmonella bacteria. So they will bind to our cells, and then they will rearrange the microfilaments in the cell so it opens up some holes in the cell membrane. And then the salmonella can just slip inside the cell. Over here on the right, we have some listeria bacteria. And remember, listeria can grow in the refrigerator and in the freezer, so they can cause some real problems with contaminated food. So what they do when they want to go to another cell is they will gather the host cell microfilaments, and because actin is one of our contractile proteins, they end up making a little rocket booster out of it. So they will gather that up and then contract the actin, and then that will shoot them out of one cell and then into the next one. So that's pretty cool. Okay, bacterial pathogens can damage host cells in a number of ways. So we're just going to talk about number 1 and 3. So the other two we won't talk about. But the first one, they can use up some of our nutrients. They can directly damage our cells, maybe by bursting them or something like that. They can also produce toxins, and sometimes they can cause some hypersensitivity reactions as well, and that can cause damage. So we'll just be talking about 1 and 3. Okay, so as far as using some of our nutrients and things that our cells need, some of the bacteria are able to produce these proteins. They're called siderophores. And the siderophores will go into our cells or into our tissue fluids, and they will bind to iron better than our cells will. And so they end up stealing iron from our cells, and the iron is going to be used by our cells and also by the bacteria as part of the electron transport chain, and we also need iron for some of the enzymes to work as well. So toxins can be produced by some of the microorganisms, and they're poisonous. And just a couple of vocabulary words. We have toxogenicity, which just means the ability of a microorganism to produce a toxin, and not all of them produce toxins, but some do. Toxemia just says that there are toxins that are in the blood, and that's not a good thing because that means the toxins can spread all over the body and cause more damage. And then an intoxication is not talking about alcohol, but it's talking about the presence of a toxin, but the microorganisms might already be gone. So the toxin has been left, but the microorganisms might be gone. So this might happen with food. So maybe Staphylococcus aureus got into your food, it contaminated it, it grew, produced some of its toxins, and then maybe you put the food into the microwave and the microwave killed the bacteria, but the toxins are still there, and if you eat the food, then you're going to get food poisoning. There are two different types of toxins that can be produced by bacteria. We have exotoxins and endotoxins. So these are really important, so make sure you know these for the test. So exotoxins, exo means to the outside, so exotoxins are made out of proteins that are going to be secreted to the outside of mostly gram-positive bacteria. They're very toxic. The most toxic exotoxin that's known is made by the botulism bacteria. So the botulism toxin, you only need like 0.03 nanograms per kilogram of body weight to kill someone. So that's a really, really small amount, so very toxic. The endotoxins are very different. They're not made out of proteins. They're made out of lipopolysaccharides instead. Remember, those are found in the outer membrane of just the gram-negative bacteria. So endotoxins are only in gram-negative bacteria. Endo means inside or within, so these are not going to be secreted by the bacteria to the outside of the cell. They're going to be released when the bacteria die. And they're not as toxic as the exotoxins are. They can still kill you, but you're going to need a larger amount to do that. Okay, and this is just a nice picture that shows the difference between exotoxins and endotoxins. So exotoxins over here on the left, you can see that these mostly gram-positive bacteria are going to be secreting these protein exotoxins to the outside of the cell, and then they will damage your cells. The endotoxins are only found in gram-negative bacteria. Remember that they're the lipopolysaccharides that are part of the outer membranes. So when the bacteria die and they disintegrate, that's going to release the lipopolysaccharides, and that's what can make you sick. This is a really good table that compares the exotoxins and endotoxins. So spend some time looking at this, and you only need to know the things that we talked about. So we're going to look a little bit more closely at exotoxins. So these are proteins that are going to be secreted by mostly gram-positive bacteria to the outside of the bacteria. They're very highly immunogenic, which means they really stimulate the immune system. So the immune system will really come after these. So because the immune system really recognizes these, we're able to produce antitoxins. So remember, antitoxins are antibodies that will bind to and neutralize these exotoxins. So for example, we can treat tetanus and botulism by giving the person antitoxins. And that will block those exotoxins that are causing the symptoms from continuing to cause problems. So because the exotoxins are really recognized by the immune system, we can also make vaccines out of them. So if you remember, we talked about the toxoid vaccines. We're just giving the person inactivated exotoxins. And the two examples of toxoid vaccines that I want you to know are tetanus and diphtheria vaccines. And then here's just another picture of exotoxins. So we have three different types of exotoxins. We have AB exotoxins, membrane disrupting exotoxins, and superantigens. The AB exotoxins, they are made out of two subunits. We have the A subunit and the B subunit. So if you look on the picture on the bottom, the B subunit is what's going to bind to our cell and will maybe open up a channel protein or something. And that allows the A subunit to go inside the cell, and the A subunit is what's toxic and will either damage or kill our cells. So there's a lot of different mechanisms how AB exotoxins work. You do not have to memorize these, but some of them will work by inhibiting protein synthesis, so maybe they interfere with the ribosomes. Some of them will block cell communication by interfering with signal transduction. And then others will actually block the release of neurotransmitters. So that's what happens with the botulism toxin. So you're not able to release acetylcholine to tell your muscles to contract, and so you become paralyzed with botulism. And then this is just another picture just showing you the bacterial cell in the top left that is making the protein exotoxin, the AB subunits, and then releasing those to the outside of the cell, and then that will bind to our cell. It's the B subunit that binds to the receptor, and then we get the A subunit inside, and the A subunit is what causes the problems for the cell. And in this picture, it's going to be interfering with making proteins. We also have some membrane disrupting exotoxins. So these are not made out of subunits, but they're going to lyse our cells by either forming some pores, so they'll form holes, and then the water will rush in, and then our cell will swell and explode. And then some of these membrane disrupting exotoxins are actually enzymes. So they are phospholipases, and what they do is they cut the heads off of the phospholipids, and that causes the plasma membrane to disintegrate and fall apart, and then the host cell will die. Here's just some examples of membrane disrupting exotoxins, and both of these are going to form pores or holes in the cells, and then the water will rush in and the cell will explode. So we have leukocidins, which are produced by some bacteria, and as you can see by the name, leuko, talking about white blood cells. And remember cide, if you remember the word like homicide or suicide, cide means to kill. So leukocidins are going to be membrane disrupting exotoxins that will form pores in phagocytes and will kill them. So that's a pretty good defense to stay alive for these bacteria. Other membrane disrupting exotoxins are called hemolysins. So they're going to poke holes in the red blood cells and destroy them. The reason that bacteria would want to lyse red blood cells is then they're able to get to all the iron that's inside that's on the hemoglobin. And we have a picture over here on the right of Streptococcus bacteria. Remember we talked about you can classify them. And so the beta hemolytic strep, they make these hemolysin exotoxins, and so they're able to lyse the red blood cells, and that's why the blood agar turns clear. The last type of exotoxins are superantigens. So superantigens are able to stimulate many helper T cells at once, even though they're not their match. So we end up with all these helper T cells being activated, releasing cytokines, and then that creates a cytokine storm. So that fills in the blank there, cytokine storm. So with all these cytokines being released, we can actually cause multiple organs to fail, and then that can kill you. So how these superantigens work is, if you look in the picture on the bottom right, we've got an antigen presenting cell, and then over there on the right, we've got a helper T cell. So the superantigen will actually form a bridge and will bind to the class II MHC protein on the antigen presenting cell as well as the helper T cell receptor. And notice that it's not binding where the specific antigen would bind, but it's able to turn on and activate the helper T cell and think it's its match, even though it's not. And so the helper T cell becomes activated and starts secreting all these cytokines. So these superantigens can actually activate up to a third of your helper T cells. So you can see how you can very quickly get too many cytokines being released. So a really good video of this is over here. There's a link on the bottom left. And an example of a superantigen is staphylococcal enterotoxin B, which is produced by Staph aureus bacteria. So it can cause food poisoning if you eat it in your food. And then if you get it into your body, if it gets into the deeper tissues, it can actually cause toxic shock syndrome. And that has been associated with a certain type of tampon use in women, but those have been taken off the market, so we don't have to worry about those anymore. Okay, and these are just some different bacteria and their exotoxins they're producing and the diseases they cause. All right, the other type of toxin that's produced by some bacteria are endotoxins. So remember, these are the lipopolysaccharides that are found in the outer membrane of only the gram-negative bacteria. So endotoxins cannot be produced by gram-positive bacteria. And if you remember the structure of the lipopolysaccharides, here's a picture on the bottom left. The part that actually makes you sick is the lipid A part of the lipopolysaccharide. And looking at our outer membrane, the lipid A part is inserted into the membrane of the outer membrane there. So what happens with these lipopolysaccharides is when the gram-negative bacteria die, they release those lipopolysaccharides, and if you release a lot of those, you can actually stimulate the macrophages to release some cytokines, which will cause inflammation and fever, and then that can lead to some bigger problems. Okay, and here's just a reminder that the endotoxins are part of the outer membrane of the gram-negative bacteria, and they're going to be released when the gram-negative bacteria die. And this is just showing you the macrophage that's eaten a gram-negative bacteria, and as it's digesting the gram-negative bacteria, it gets exposed to those lipopolysaccharides, which stimulates the macrophage to secrete some cytokines, which go to the hypothalamus, where the thermostat is, and resets that and causes a fever. And so we do see fevers with endotoxins, but not with exotoxins. You don't need to know this flowchart, but it's just showing you that if you have a release of a large amount of endotoxins, if you have a lot of gram-negative dying all at once, you can get some pretty serious problems being triggered in your body. So besides the fever, we can also get what's called disseminated intravascular coagulation, or DIC. And DIC just means that the clotting factors in the blood will get triggered and you start clotting your blood inside your blood vessels. So disseminated just means widespread, and then intravascular just means you're getting these clots forming inside your blood vessels. And that's bad because these clots will then lodge in capillaries and then block blood flow to whatever's on the other side. And then eventually you run out of clotting factors and then you can't stop bleeding and you can actually bleed out. And then the other thing that the endotoxins can cause is septic shock. And shock is just where your blood pressure gets too low and you're not able to get enough blood to your organs, and so then your organs start to fail. So we can have a pretty high death rate if we do have patients that are getting this DIC and septic shock, and that's pretty hard to stop all that. Okay, so these virulence factors that we've been talking about, they're coded for by genes that are located on plasmids. And remember that plasmids, we can share copies of those through horizontal gene transfer with other bacteria. So we can actually spread some of these virulence factors around, and these bacteria can share their great ideas. Another thing that we talked about before is phage conversion. So a good example of this would be the bacteria that cause diphtheria. So if the bacteria that causes diphtheria has not been infected by a bacteriophage, it will not make you sick. But if it has been infected with a particular bacteriophage, the prophage, the viral DNA, gets inserted into its chromosome, and the viral DNA actually codes for a toxin that causes diphtheria. So that would be an example of phage conversion. So the bacteriophage, the virus, will convert the bacteria from not causing disease to being able to cause disease by producing this toxin. All right, just looking at some pathogenic properties of viruses, they are able to hide from our immune system by multiplying inside the cells. And remember that they will adhere to our cells, they will bind with their spike protein to our host cell receptors, and then they'll get inside that way. And so here's showing an HIV virus. So viruses can cause some cytopathic effects on cells. So some of them will outright kill the cells, but in others, if they have an envelope, they're going to leave the cell by budding, and so they're going to damage the cell and kill it slowly, but we're going to see some effects that tell us that that cell is infected with the virus. So there's a lot of different cytopathic effects, but the ones I want you to know are the inclusion bodies, syncytia, and then transformation. So remember we talked about viruses being able to transform normal cells into cancer cells. So a good example of that would be the hepatitis C virus can do that. And then here's just some cytopathic effects. This is the rabies virus over on the left. And this is in the brain. We've got some of our neurons here. And you can actually see an inclusion body, which is this little dark staining structure inside the neuron. And an inclusion body is just a virus factory. So you can actually see where they're putting all the pieces together with the capsids and putting the viral nucleic acid inside, and so that's just a virus factory. Other viruses, like for example the measles virus, when it wants to infect the next cell, it actually, instead of going outside the cell where it's more susceptible to be grabbed by the immune system, it actually will cause that infected cell to fuse with the next cell. And so these cells just all fuse together as the virus spreads and infects other cells. And so we end up with this gigantic cell that has multiple nuclei in it, and that gigantic cell is called a syncytium. Because remember, syn means together and cyte means cell. So you've got all these cells that are fused together. So if we see that, then we would be suspicious of measles virus. Okay, so looking at fungi, some of them are able to produce mycotoxins. And the mycotoxins, we often find these as contaminants of food crops. So there's a mold called Aspergillus that will produce some aflatoxins. And the aflatoxins are bad because they're carcinogenic, so they can actually cause liver cancer. There's another fungus that will produce what's called ergot. And the ergot, this is showing you some rye on the bottom, and so these kind of spiky structures that are sticking out, that's the ergot. So the ergot are actually reproductive structures that the fungus will use to produce spores and then spread to other plants. So the problem is that the fungus will produce a mycotoxin that's similar to LSD, and so it actually causes hallucinations. So that would not be so good to have in your cereal. Okay, looking at some of the protozoans, for example, over here on the right, we've got a picture of malaria. So the malaria will actually divide and multiply inside the red blood cells. And then when they've filled up the red blood cell, they will burst the red blood cell and then go find other red blood cells to infect. And the bursting of the red blood cells, that's what causes the symptoms of malaria. Looking at algae, some of them are able to produce some neurotoxins. So for example, sometimes when we have red tides, some of the species of algae that are in there produce these neurotoxins, and they get into shellfish because shellfish are filter feeders, and those neurotoxins will lodge in there. They don't hurt the shellfish, but then if you eat the shellfish, then that neurotoxin will actually paralyze you. So saxitoxin can cause paralytic shellfish poisoning, and you don't have to memorize that. Okay, and then once they've had a party in your body, damaged your cells, caused disease, made you sick, now they want to exit and find another host. So the portal of exit is often going to be the same as the portal of entry for a particular microorganism. So for example, cold viruses entered into the sick person through their respiratory system, and they're going to exit the same way and then enter the respiratory system of the next person and infect them. And again, just to remind you where we went in this chapter, we talked about how microorganisms get into your body through the 3 portals of entry. We talked about how they adhere or stick to your cells. And then we talked about how they could penetrate deeper into your body, evade your immune system. They will damage your cells, which will cause our symptoms of disease. And then once they're done, they will exit the body through these portals of exit and go find another host.